So the function of the eye is to receive light and refocus the light onto the retina at the back of the eye. Know the structure of the eye and being able to label it, and also the function of parts of it. So particularly know your lens, suspensory ligament and ciliary body, the role of the iris, letting the light or adjusting the amount of light that can pass through the pupil. Then the light is focused onto the retina, the fovea, the area that has high concentration of cone cells, the sclera being the tough muscle or tough outer layer of the eye. So read over the notes about the structure of the eye. So controlling the amount of light that enters the eye is under the control of the iris. So in dim light, the key things that will happen, the radial muscles will contract. So they are the long muscles radiating out in the iris. The circular muscles relax. This causes the pupil to dilate, or the diameter of the pupil to increase, and that allows more light to enter the eye. So you can see in the diagram showing you that the radial muscles contracted, so they are getting shorter. The circular muscles are relaxed, causing the diameter of the pupil to increase. In bright light, really the opposite is happening. So our radial muscles relax and become longer. The circular muscles contract. That causes the pupil to constrict and the diameter to get less. So less light will enter into the eye, less likely to damage the retina. Accommodation. This is where light is focused onto the retina, and it is accomplished by the lens changing in thickness. So the lens is elastic and its shape can be changed when the suspensory ligaments attached to it become either tight or taut or loose. The changes are brought about by the contraction or the relaxation of the ciliary muscles. So near accommodation is what we would call focusing on an object that is close. So a number of things happen. The ciliary muscle contracts. This causes the suspensory ligaments to slacken, which in turn means that the lens becomes thicker and fatter. That means that the light gets refracted more or bent more as it enters. So therefore the image will form at the back on the retina. Far accommodation, looking at an object far away. Again, that would be relaxing. So the ciliary muscles relax, so they're not having to work. That then causes the suspensory ligaments to become taut or tighten, which pulls the lens and makes it become thinner. This means that there's less bending or refraction of the light, and again, an image then forms on the retina. So be able to describe the changes that occur for near accommodation and far accommodation. The retina, as we've mentioned, are made up of the rod and cone cells. So your rod cells and cone cells contain optical pigments that absorb the light and break down. So the rod cells contain the pigment rhodopsin and the cone cells contain iodopsin. The breakdown of these pigments results in a generator potential being produced. If the threshold value then is reached, then the bipolar neuron that is linked to the rod cells or the cone cells will be depolarized and an action potential is produced. The pigments within the receptors are broken down by different conditions, which we're going to look at. So our rod cells. So rhodopsin is very sensitive, so it will break down in dim light. So we would say that rod cells have high sensitivity. And as it says there, sensitivity refers to the amount of light that is required to stimulate the receptor and break down the pigment that will in turn create a generator potential. The rhodopsin breaks down to opsin and retinal, and then your mitochondria that are found in the rod cells, their function is to release ATP for the resynthesis of the rhodopsin. Benefit of your rod cells is that they allow humans to distinguish between light and dark objects when light is very dim, but they do not allow humans to see in colour, so they are described as being monochromatic. Our cone cells contain the pigment iodopsin. It is less sensitive, or it has low sensitivity, because it will only break down in bright light. But they are sensitive to different wavelengths of light. So there are 3 types of cone cells that we would find: blue, green and red. So they're described as having trichromatic vision. So our red sensitive cones will be sensitive to wavelength of light that are corresponding to the colour of red, blue sensitive cones to blue light, green sensitive cones to green light. So they have low sensitivity. Visual acuity. So acuity is the ability to distinguish between two separate points, particularly two points that are close together. So it's similar to the term resolution. So if you were at the opticians and you were able to read the small letters at the bottom of the board, then you would be said to have high visual acuity. Think of it that way. So our rod and cone cells, the receptors, are hit by light, and if you remember, it is an inverted image, so the light has to pass by the ganglion cells, the bipolar neurons, before it hits your receptor cells. And then behind that you have your sclera, the back of the eye. So the rod cells and the cone cells are hit by the light rays. They become stimulated. Any that aren't hit by light rays then don't become stimulated, so they're unstimulated. The receptor cells, as you can see here, the rod and cone cells, then synapse to bipolar neurons, and then the bipolar neurons are connected or synapse with ganglion cells. The ganglion cells have long axons that extend to the optic nerve, which then is directly connected to the brain. So we say that rod cells have low visual acuity. So this means that multiple rod cells will synapse with a single bipolar cell, and this is known as retinal convergence. In turn, those multiple bipolar cells synapse with a single ganglion cell, so the brain is not able to interpret which impulses are sent by which specific rod cells. Retinal convergence allows the generated potentials from individual rod cells to combine together, so this is our summation, or the spatial summation, and it allows it to reach the threshold value, so an action potential can be generated in the bipolar neuron. So retinal convergence will ensure that action potentials can be fired off in that dim light because of the summation. And as you can see in this diagram, looking at the rod cells, you can see that 3 rod cells are synapsing to one bipolar neuron, and then it's synapsing with another set bipolar neuron to a single ganglion cell. So the information is getting diluted down each time as it synapses. Our cone cells, they synapse with a single bipolar cell. The single bipolar cell synapses with a single ganglion cell. So if we go back to the previous slide, we can see that on the cone cells on the right, each has its own bipolar neuron, and each bipolar neuron synapses with its own ganglion cell. So this is going to have much better acuity than our polar cells. So if 2 cone cells were stimulated to send an impulse to the brain, then it would be able to distinguish the 2 different spots of light. And because our cone cells detect either red, green or blue, then the information that the brain would be receiving would be telling about what amount of red cone cells have been stimulated, green or blue cells have been stimulated. So the diagram at the bottom here shows you 1 cone cell has just been stimulated, so the brain's going to perceive that as 1 spot of light. 2 cone cells stimulated, the left, one on the very left and one on the very right. Again, 2 spots been seen separately. And the same for the diagram C as well. 2 spots will be seen separately. So you may be shown a diagram showing you the layout of the rod and cone cells in the eye. So our peripheral vision at the very edges, so if you're looking at the 70 degrees on the left hand side and the 80 degrees on the right hand side, we can not see colour because there's no cone cells there, but we can see monochromatic shades of black and white because of the rod cells. At the blind spot, there are no cone cells or rod cells, so we will not be able to perceive anything. And cone cells, the highest numbers are at the fovea, and that's where they are found. Other things to think about, that this graph shows you for the left eye. As you can see, there are more rod cells closer to the centre of the head on the right side of the blind spot. But there are still rod cells to the left of the blind spot, but their numbers get lower as you move towards the 70-degree angle. And this lower amount still allows for that peripheral vision. If you were shown a graph for the right eye, then it would look the other way about. So more rod cells being on the towards the left side, towards the centre of the head. Binocular vision or stereoscopic vision. So having two eyes gives you this stereoscopic vision, and that the benefit of this is that it allows accurate judgment of distance, and it allows the ability to form three-dimensional images. Predators, their eyes are out front so that they have good judgment of distance and 3D vision, whereas preys, their eyes are at the side of their head because it's more important for them to have a wider field of view so that they can detect the predators. So that is the eye.